Scientists just discovered something that changes everything about plants. You talk to your plants, maybe not out loud, but you move them toward the window. You check the soil. You notice when they look wrong. You've always sensed that something is happening in those leaves, something responsive, something almost aware. It turns out you were right, and what researchers have been quietly documenting over the last 2 decades goes far beyond anything most people were taught in school. Because plants are not passive, they are not silent, and they are not separate from each other the way they appear. This is not a metaphor. This is biochemistry. And once you understand what's actually happening inside and between plants, the way they communicate, the way they respond, the way they remember, it changes how you think about everything you grow, everything you eat, and everything your great-grandparents knew that somehow got left behind. This is where the real story begins. What people see, surface reality. Most people see plants as a category of thing. Living, yes, but passive, rooted in place, unable to respond, unable to signal, unable to protect themselves without human intervention. That view shaped everything: how we farm, how we garden, how we designed the food system that most of the developed world now depends on. The logic followed naturally. If plants can't respond to threats, we respond for them. If they can't communicate a problem, we monitor for them. If they can't protect themselves, we spray for them. Pesticides, fungicides, synthetic fertilizers, monoculture at scale, all of it built on an assumption that is now being quietly revised by the researchers who study plant behavior at the molecular level. The assumption was, plants are chemical machines. Put in water, sunlight, nutrients. Get out growth, fruit, seed. That's not wrong, but it is, it turns out, severely incomplete. And that gap between what plants were assumed to be and what they are actually doing is one of the more significant blind spots in modern agriculture. What's actually happening? In 1983, 2 researchers working independently, Jack Schultz at Dartmouth and Ian Baldwin, then a graduate student, published findings that the scientific community largely dismissed at the time. They had observed that when caterpillars attacked maple and poplar saplings, neighboring trees that had not been touched began producing elevated levels of tannins and other chemical compounds in their leaves, compounds that make leaves less palatable, harder to digest, effectively toxic in higher concentrations to the insects causing the damage. The untouched trees were responding to a threat they could not see, could not feel, and were not physically connected to. Schultz and Baldwin proposed that the damaged plants were releasing airborne chemical signals, volatile organic compounds, that the neighboring plants were detecting and responding to. The response from the scientific establishment at the time was skeptical. The idea that plants could warn each other seemed too close to anthropomorphism, too close to attributing a kind of intention to organisms that were not supposed to have any. 40 years later, the mechanism has been mapped, the chemistry confirmed, and the field of plant signaling has grown into one of the more active areas of biological research. Here is what is now understood. When a plant is damaged by an insect, a pathogen, physical injury, even intense UV light, it releases a class of chemicals called herbivore-induced plant volatiles. These compounds travel through the air. Neighboring plants detect them through chemical receptors in their leaf surfaces, and within hours, those neighboring plants begin producing their own defensive compounds, effectively priming their immune systems before the threat arrives. This has now been documented in dozens of species: corn, tobacco, tomatoes, sage, beans, lima beans, cotton, barley, wheat. The signal travels. The message is received. The response is real. This is not a metaphor for communication. It is communication. The medium is chemistry rather than sound or light, but the function, information transfer that changes behavior in the receiver, is the same. And this is only one layer of what's been found. Why this changes things. Here is the part that most people miss when they first encounter this research. The discovery that plants signal each other above ground was significant, But it was the work happening below ground that began to reshape how biologists think about plant life entirely. In the 1990s, forest ecologist Suzanne Simard, working in the old-growth forests of British Columbia, began investigating something foresters had noticed but not fully explained. When large, established trees were removed from a forest, the seedlings around them often declined, not immediately, not dramatically, but measurably, as though something had been disrupted beyond just canopy cover and shade. What Simard found, through a series of careful isotope tracing experiments, was a network. Beneath the forest floor, the roots of trees are connected to fungi, mycorrhizal fungi, that extend through the soil in thread-like structures called hyphae. A single fungal network can extend for miles. It connects dozens, sometimes hundreds of individual trees. And through this network, trees exchange carbon, nitrogen, water, and chemical signals. Not randomly, selectively. Simard's research showed that larger, more established trees, what she called mother trees, actively transferred carbon and nutrients to younger seedlings in low-light conditions. The seedlings were receiving more than they could produce themselves. The network was compensating for their deficit. When mother trees were stressed, attacked by insects, drought, disease, they increased the transfer of carbon to their root systems and to connected seedlings, a kind of pre-death distribution of resources to the next generation. This research has been peer-reviewed, replicated in multiple forest systems across the world, and the basic mechanism, carbon transfer between trees via mycorrhizal networks, is now accepted within the relevant scientific literature. What is still being studied, still debated, is the degree of intention, coordination, or selectivity involved. What is not debated is the transfer itself. The forest, it turns out, is not a collection of competing individuals. It is, in significant part, a cooperative network. And that network is managed by fungi, organisms that take a small percentage of the carbon they facilitate in exchange for the service they provide. A system that has been running, largely unchanged, for approximately 450 million years. This is the part that tends to stop people. Why this changed, and what got lost. Before the mid-20th century, most agricultural soil contained rich, diverse communities of mycorrhizal fungi. Soil was alive in a way that has no direct analog in most modern agricultural systems. A teaspoon of healthy traditional farmland soil, the kind of soil that would have been farmed using methods passed down through generations before synthetic inputs became available, contains somewhere between 100 million and 1 billion individual bacteria, thousands of fungal species, nematodes, protozoa, arthropods at the microscale, a food web of extraordinary complexity, all operating within a few inches of topsoil. The mycorrhizal networks were part of that system. They connected crops to each other and to the broader soil food web. They extended the effective root reach of plants by factors of 10 to 1,000. They made phosphorus, zinc, and other minerals biologically available in ways that plant roots alone cannot achieve. Then came the Green Revolution. Between roughly 1950 and 1970, synthetic nitrogen fertilizers became widely available and rapidly adopted across industrial agriculture. Yields increased significantly. Famines that had seemed inevitable were avoided. The achievement was real and substantial, and it is not the purpose of this to dismiss it. But synthetic nitrogen, applied at scale, does something to mycorrhizal networks. When nitrogen is abundant in the soil, readily available, no longer needing to be sourced through fungal partnerships, plants reduce or eliminate their investment in mycorrhizal relationships. There is no metabolic incentive to feed a network you no longer need. The fungi decline, the networks thin, the cooperative infrastructure built over millions of years begins to recede. Add to this the effect of many synthetic pesticides and fungicides on soil microbial communities, and the compaction effect of heavy agricultural machinery on the soil structure those networks depend on. And what emerges is a picture of a system that was progressively simplified. Higher yields, shorter supply chains to famine, but also soil that increasingly functions as a growing medium rather than a living system, plants increasingly isolated from each other, dependent on inputs rather than networks. Crops with measurably altered nutritional profiles compared to their ancestors. That last point has been studied. A widely cited analysis published in the Journal of the American College of Nutrition in 2004, examining USDA nutritional data for 43 common vegetables and fruits from 1950 to 1999, found reliable declines in protein, calcium, phosphorus, iron, riboflavin, and vitamin C. The researchers attributed this primarily to the shift toward varieties bred for yield and appearance rather than nutritional density, and to the soil conditions those varieties were grown in. This is not a fringe finding. It has been replicated in subsequent studies using different datasets in the UK, Canada, and Australia. The food looks the same. In many cases, it yields more per acre than it ever has. But what's inside has quietly shifted. The consequence: clarity, not fear. This matters for a reason that is worth stating plainly. If the signaling systems between plants were disrupted, if crops grown in chemically simplified soils without mycorrhizal networks are essentially cut off from the cooperative chemistry that wild and traditional agricultural plants had access to, then the food they produce may be chemically different in ways that go beyond the decline in minerals already documented. There is emerging research on this. It is early. It is not settled science. But there are studies examining whether plants grown with intact mycorrhizal communities produce different secondary metabolite profiles, the polyphenols, flavonoids, terpenes, and other compounds that are not strictly nutrients, but that have significant biological effects in humans. Anti-inflammatory compounds, compounds that influence gut microbiome composition, compounds that, in the fermentation research discussed elsewhere in this space, become far more bioavailable when properly processed. The hypothesis, stated carefully, is that the cooperative chemistry between plants and their networks may influence not just yield and resilience, but the full biochemical richness of what those plants contain. If that hypothesis continues to hold up under investigation, it would mean that two tomatoes, visually identical, grown in fundamentally different soil systems, could have meaningfully different effects on the person who eats them. This has been debated. There is limited data on the full scope of it. But the directional evidence is consistent. Soil health affects plant chemistry. Plant chemistry affects human biology. And the chain connecting ancestral food to ancestral health runs in significant part through soil systems that most modern food production has quietly dismantled. This is the part that tends to land differently once you've seen it. The pattern and why it's not new. This is not the first time this kind of shift has happened. The pattern is familiar if you look at it across enough domains. A complex, evolved system, one that has been functioning and adapting for millions of years, gets replaced by a simpler, more efficient, more scalable version. The immediate results are positive. Yields rise, costs fall, access improves. The case for the new system is compelling and largely accurate. But the complex system was doing things that weren't fully understood at the time, things that took decades to document, things that didn't show up in the metrics being measured. This happened with fermented food. Traditional cultures preserved vegetables, grains, legumes, and dairy through fermentation, a process that, as we now understand, dramatically increases the bioavailability of the nutrients those foods contain and produces compounds like GABA, short-chain fatty acids, and bacteriocins that have significant effects on gut and nervous system function. Industrial refrigeration made fermentation unnecessary for preservation. The traditional practice declined. The biochemical benefits went with it. Nobody decided to remove those benefits. They were simply not fully documented at the time. They were assumed to be incidental to the real purpose of the preservation method. This happened with whole grain milling. Traditional stone milling produced flour that retained the germ and bran of the grain, the parts that contain the majority of the grain's mineral content, fiber, and fat-soluble vitamins. Industrial roller milling, developed in the 1870s, produced flour with a longer shelf life, a finer texture, and a higher proportion of endosperm starch. commercially superior, massively adopted. What was removed in the process was not fully understood until the deficiency diseases that followed, pellagra, beriberi, began appearing in populations that had shifted entirely to refined grains. The response was fortification, adding back a small selection of the removed nutrients synthetically. But fortification replaces a fraction of what was removed and does not replicate the matrix, the way those nutrients exist in relationship with each other and with the other compounds in the whole grain. The same pattern. Each time, simplification for scale, followed by the slow emergence of what was lost. What the researchers are actually saying. It is worth being precise here, because this research is sometimes distorted in both directions, either dismissed as fringe science or overstated as a revolution. What the mainstream plant science literature now accepts: plants exchange chemical signals through airborne volatile compounds. This is established. The mechanisms are mapped. The responses in receiving plants are documented. Plants connected through mycorrhizal networks exchange carbon, water, and chemical signals. The carbon transfer is confirmed. The full scope of chemical signaling through the network is still being studied. Soil microbial diversity affects plant chemistry, resilience, and nutritional content. The directional relationship is established. The magnitude of the effect in specific contexts is still being quantified. What is still being studied, still debated, still incomplete? The degree to which plants exhibit something that could be called learning or memory. Early studies suggest some capacity for habituation to repeated stimuli, but this is a contested area with significant methodological debate. The precise relationship between mycorrhizal network connectivity and specific secondary metabolite production in crops. Early research is suggestive but not conclusive. The full implications for human nutrition of the shift in soil systems over the last 70 years. The directional evidence is consistent, but the magnitude is not settled. This is how science actually works. It moves in increments. It revises. It holds some things firmly and others loosely. The firm things here are firm enough to be worth understanding. Practical shift. What this means in practical terms. None of this requires a complete overhaul of how you eat or garden or think about food. But there are some things worth considering, given what the research currently shows. If you grow food, even in containers, even on a balcony, soil life matters in ways it wasn't thought to before. Mycorrhizal inoculants are now commercially available. The research on their effectiveness varies by context, but in soils depleted by repeated synthetic fertilizer use, restoring fungal communities has shown measurable effects on plant health and nutrient uptake in multiple studies. Avoiding synthetic fertilizers, particularly high nitrogen formulations, in your own growing system, even partially, allows existing mycorrhizal networks to persist and develop. The plants work harder for their nutrients and, in doing so, produce different chemistry. If you source food, the research on soil health and nutritional content is an argument for produce grown in living soil systems, organic, regenerative, or small farm grown, not as a moral position, but as a practical one. The evidence that soil system affects end nutritional profile is consistent enough to take seriously. The fermentation piece connects here directly. If plants grown in richer soil systems produce fuller secondary metabolite profiles, and if fermentation unlocks those compounds and increases their bioavailability by factors of 2 to 5, the combination is not trivial. Traditional food systems that used both living soil agriculture and fermentation preservation were not doing so by accident. They were preserving a chain of biochemical benefit that, at each link, has since been partially broken. Reconstructing that chain is not complicated. It does not require specialist knowledge or unusual ingredients. It requires understanding what the chain was and why each link mattered. That understanding is what this kind of research slowly makes available. The bigger pattern. Zoom out far enough, and what you see is a consistent theme across the last century of food system development. Efficiency, scale, and shelf life were optimized. Measurable outcomes, yield per acre, cost per calorie, time to spoilage, were tracked carefully. The things that were not measured: soil microbial community density, plant secondary metabolite profiles, the full scope of what fermentation produces, the relationship between mycorrhizal network integrity and crop chemistry, were not lost through malice. They were lost through the ordinary process of optimizing for what you can measure, while not yet understanding what you're not measuring. The researchers now beginning to map what was simplified out of the food system are not critics of the choices made. They are scientists working with tools that did not exist when those choices were made. What they are finding, incrementally, across multiple independent research lines, is that the systems that were simplified were doing more than the simplification preserved. The plants were not passive. The soil was not just substrate. The food was not just calories, macronutrients, and a handful of vitamins. And the traditional practices, the fermented foods, the living soil methods, the polyculture planting systems, the whole grain processing, were not primitive approximations of what we now do better. They were, in many cases, doing things we are only now beginning to understand, and, in some cases, trying to reconstruct. That is not a comfortable conclusion. It does not fit neatly into the narrative of progress as a straight line. But it fits the data. Here is where this matters practically. The gap between what plants are actually doing and what the food system was designed around is a gap that shows up, eventually, in the people eating from that system. Not dramatically, not in ways that are easy to attribute, but measurably, consistently, across enough different research lines to constitute a pattern worth paying attention to. The researcher who studies mycorrhizal networks, and the researcher who studies plant volatile signaling, and the researcher who studies nutritional content decline in modern produce, and the researcher who studies traditional fermentation are not, for the most part, talking to each other. They are each working on one part of a much larger picture. The picture they are collectively building is of a food chain that was more sophisticated than understood, simplified in ways that were rational given what was known, and now beginning to reveal the cost of that simplification through a slow accumulation of evidence across disciplines. You do not have to wait for a consensus statement to act on this incrementally. The moves are small. Grow something in living soil, ferment something, source food from systems you understand. Reduce dependence on inputs by learning what the inputs replaced and whether the replacement was equivalent. The knowledge to do this is not new. It is, in many cases, the knowledge your grandparents' generation still held, and that the generation after them was told was no longer necessary. That knowledge turns out to have been more necessary than it appeared. And this same pattern, the thing that was replaced being more important than the replacement suggested, appears in more places than most people have had reason to look. That is where this series continues, because once you see the pattern here, you begin to recognize it elsewhere. And once you recognize it elsewhere, the picture becomes considerably harder to ignore.